are the turtle ants named for, is that the shape of their abdomens, or is it that they're living on the ground, or
So these are actually all arboreal.
they're
And so
arboreal? Got it.
interestingly, you can kind of see in this picture, but they have like a shield type head where
Mm,
they can tuck their antenna inside, because that's one of the most delicate parts of their body, right? It's where they do all of their communication. It's what they're sort of feeling the environment. So they can tuck them under these little ridges to protect them if there's an intruder or if they're engaged in a battle. So the fact that they can sort of tuck things under a shell-like, parts of their body is where they get the name turtle ants.
cool. So you had one more intriguing ant photo here. There's some kind of behavior going on here for sure.
Yeah, for sure. So that is the bullet ant, Paraponera clavata.
Ah,
And you can see that she's captured some sort of a prey item and is taking it back to the nest to feed to the young. So interestingly, when ants are adults, you can see how she has such a skinny little narrow waist. It almost
yeah.
looks like a tiny straw connecting the thorax with the rest of the abdomen. And in this case, because it's so tiny, they can't eat solid food items. So they can drink any kind of sugar source or liquid food source
Huh.
they find. But the reason they always see adult ants dragging these prey items back to the nest is to feed to the larvae. So the larvae don't look like adult ants yet. They kind of look like a fly maggot. They're essentially just a sac with a mouth, and they are the ones that need all that protein to continue to grow and develop.
That's man, that's crazy.
Yeah, and so this is
So
actually
pinched that they can't get solid food through there.
right, but then that pinch allows them to have flexibility so that they can bring the sting all the way forward under their bodies, right? So as they're stinging a prey item or an intruder, they actually can get at them in any angle. And if they were sort of like the grasshopper prey, right, that one has almost no flexibility. It couldn't move the tip
Mhm.
of its abdomen in almost any direction. And this ant in particular is called the bullet ant because its sting is so painful. They say it's like you've been shot by a gun. So it's also called the 24-hour ant because the pain lasts for 24 hours.
Ah, and these ants are in Amazonia. Is that right?
That's absolutely
Yeah,
right. They're found from Central America all the way through South America.
that's that's remarkable. Okay, so you've convinced me. Ants are ants are really interesting. You sent this shot of. So I think one of the interesting things is that you know you've got these big questions and this curiosity you have, and you found a group that really lets you explore these things. Um, but I think it's also interesting that you work in the field. You also have a sort of familiar looking wet laboratory that includes molecular analysis, and then there's collections. So maybe take a couple minutes to talk about that. So here's you're collecting ants somewhere, I take it?
Yeah, absolutely. So I've been very fortunate that I've been able to travel around the world collecting ants. I really love working in the tropics. It's where you have the most ants. That's also very hot and sweaty and familiar, like where I grew up, unlike upstate New York here. And so one of the things I love the most about participating in research is that I get to go out in the field and watch these animals carrying out their behaviors, see exactly where they live, trying to understand when you have species co-distributed, what's actually there, what kind of partitioning of the habitat, so which ones are arboreal, which ones only live in one kind of stick versus another, who lives in the leaf litter, trying to understand the interactions, who's competing for food. And a lot of the questions I ask scientifically actually come from observations in the field. I might get an idea almost like a hunch, which then leads me down the road to start to ask questions or hypotheses. Now, once I have those sort of hypotheses in my head and I've collected the specimens in the field, that's when I take the opportunity to bring those specimens back to Cornell. And in many cases, what we're doing is a multitude of things. The first, as you alluded to, is I'm actually very involved in collections-based research. And what that means is that we keep specimens in these museum cabinets and these museum drawers so that we can look at them now, but scientists can look at them for hundreds of years. And what those specimens on those little pins allow us to do is to look at the anatomy. We can ask questions about their distributions. And they
And
serve.
where and when they were collected is key.
Absolutely. You know, one of the things that I get asked a lot, my entire career, I've been embedded in a museum, either a standalone museum or a university-based museum. And people often come and they'll see drawers full of, you know, one kind of insect or one kind of organism. And people often ask, when will we stop killing things to put things in museums?
Yeah.
And the answer is never. Because it's not just that we need to know that something ever existed. We need to also understand, has it changed through time? Not just in its distribution, but we can ask questions about its morphological diversity or an anatomical diversity. We can ask questions about its genetic diversity. And unless we have the material to see how things are sort of changing or not changing, we can't actually make statements. And I think one of the most sort of important ways that we're addressing questions in science right now is through the lens of climate change. We can actually demonstrate that some animals or organisms are shifting their distributions. We have, you know, 100 years of data of these specimens and where they were found, and we can show that they in many cases are no longer where they used to be, and in some cases are being pushed into new areas if they're not driven to extinction. So museums are really important. So we bring them back, we look at them on the pin. And as you know, you already mentioned, most of my work is sort of based on DNA and genomics. And even though that means I'm going to take one individual from a colony and grind it up to do that molecular work, I also need to have a voucher for that, because you could imagine I could say, I think I collected Cephalotes varians, and someone else could say, you don't know Cephalotes varians from Pseudomyrmex gracilis. I don't trust that DNA sequence you put up on the you know, shared servers.
Yep.
On these public databases. So if I say, well, no, this individual ant came from the same exact collection and it's serving as the voucher for all that molecular data, people can actually go and check. Even not just now, even in a few hundred years. So if the taxonomy changes or if somebody starts to question whether I knew one ant from another, they can actually go and verify that.
Yeah, so super, right. So these collections are of historical significance, but for questions that you're asking right now. And climate change is an obvious one, but there's all sorts of trend data that you might be interested in, including, you know, like in our project for the Half Earth project, we're publishing lots of data on the ranges of, you know, and it's just surprising how incomplete the data is on where even known organisms live. And then on top of it, their ranges can be shifting. So
that's absolutely true.
Yeah.
And as you know, that a bunch of us in the ant community are participating in the Half Earth project to try to get ants as one of those focal groups where you can actually explore the data.
That's great. So maybe you can give us a little insight into where is your current research? What's the sort of most exciting thing that you're working on right now?
Yeah, so I'm working on a couple of projects. One of which is that because a lot of the research I've done is focused on sort of the DNA and genomics of the ant host, and at the same time, sometimes we're looking at the microbial communities found within their guts. That often means I'm doing careful dissections of ants to sort of get at the different gut compartments. And one of the things I had noticed, because I've dissected, you know, hundreds to 1,000 ants probably at this point, I noticed that the ants.
You use little tiny needles to.
Yeah, so we use essentially little watchmaker forceps. So, you know, and I have to do everything under a microscope. It's a tedious, very difficult work, which means you can drink no coffee that day, which makes me very grumpy. But
that's a bad day.
It is a bad day, but it's so much fun. So one of the things I had noticed as I was doing this is that the ants with the thickest or toughest cuticles were either predatory species or they were entirely herbivorous, but we knew they had bacteria in their guts and that were likely involved in some kind of nutritional upregulation. So with some colleagues, we've already been able to demonstrate that the bacteria in the guts of some of these herbivorous ants do improve the nutritional sources or provide amino acids to the host. But we didn't know exactly how the host was using those amino acids that were synthesized by the gut bacteria. So just like we have helpful bacteria in our guts that we need to process food, it turns out lots of kinds of organisms do, and some ants are among them. The herbivorous ones are, in fact. So this led me down the path. Well, if these bacteria are synthesizing amino acids, maybe that's getting incorporated into the cuticle. So what we did is a whole series of analytical chemistry along with some microbial ecology, and we're able to demonstrate that the bacteria inside the guts of these ants are enriching the amino acids that are getting incorporated into the cuticle, and that's what makes the herbivorous ants have tough cuticles.
It's because their symbionts allow them to make tough cuticles.
Yeah,
Huh.
so it's really cool.
Wow.
So that's one project we've been working on. Another project I've been working on with a postdoc and some collaborators is we've been actually trying to ask the questions about using those museum collections and the data and resampling, modern resampling, to understand what happens to native ant communities through time. And so in this project, we've been looking at native ants across Florida. And we've been able to demonstrate, and maybe not surprising if you know a lot about Florida, is that a lot of invasive ants have come in. Unfortunately, they've been pushing the native ants out. And that one of the questions people have often worried about is when we have these sort of museum collections, unfortunately, they can come with bias in how they were collected. We know that most collectors are lazy. You get out of your car, you sample along the road, right? Or we also know that some collectors were super fastidious and spent hours out every single day, where others picked up a handful here and a handful there.
Yeah.
So we weren't certain whether we could actually use museum data to ask these kinds of questions.
So,
So
so absence data is really tricky when you're
exactly, and we rarely have absence data. So we've been able to go in and use a bunch of ecological modeling and explicit tests,
sampling.
and to ask the question, are these data valid for these kinds of questions? And it looks like if you have enough of it, the data overwhelms the noise. And so it doesn't matter as long as you have, probably thousands of records, you're going to find the pattern there. So it's been really interesting to sort of say, not just that we can document how things are changing through time, but we can actually advocate that museum collections are going to be an enormously important resource to understand the impacts of invasive species.
Mm. The Half Earth Project and the sort of work you do, we're all concerned about threats to biodiversity and the current status to biodiversity. From your lens of looking at things through ants, how are the ants doing? Are the ants going to inherit the earth, or is it, is there a story there that you can tell about the state of the biodiversity of ants?
Yeah, it's complicated, as you can well imagine.
Yeah.
Unfortunately, humans are having tremendous negative impacts on all biodiversity, and ants are among them. We do know that many ants are quite resilient and are likely to be able to put up with the level of disturbance that we create. Unfortunately, most of them are the invasive species
Right,
that are
as you were
which
just talking about in Florida. Yep.
exactly. And so we might still have species around, but they are certainly not going to be the native ones that have been part of those habitats and communities and ecosystems for millions of years. And so it does lead the question of what's going to happen in those ecosystems, because in many cases ants are among some of the most important ecological contributors to the habitats that they're found in. And so I think anything we can do to promote the maintenance of these native species is super important.